So let me come over here so we can see the entire. You want lights on or lights off?
Lights off.
Lights off? Okay. So, did we cover this effectively? Does everybody understand what's going on? Okay. Let's go to the next question then. Or second question. This is citrate test. So remember, if you know the name of the test, a lot of times you know what the substrate is, right? What's being broken down. So here, why is the citrate test called the defined medium? Because you know exactly what's in it. Okay? What's the indicator in this medium? What's the pH indicator? Bromothamol blue. Bromothamol blue. A blue color indicates what pH? Huh? Alkaline. Basic. A positive citrate test is what color? Blue. Positive means that the microbe can use something as its sole source of something else. Yeah.
Use citrate as its source of carbon?
Correct. That's a beautiful job.
Citrate?
Beautiful job. Citrate as its sole source of carbon. When that happens, ammonia gets released into the medium and the pH goes up. Blue indicates alkaline because the pH has gone up. Okay? Ammonia gets released in anything, the pH is going to go up. Okay? Anybody have questions about this? How do you inoculate this? How are you going to say that in words? SSSB. SSSB is a fair answer here. You don't have to say streak the slant, stab the butt. You can just, and please don't say just stab the butt. That would be wrong. Right? But SSSB is fine. Okay? Here we go. This is the MR and the VP, but we're doing the MR first. What reagent is added to the medium after incubation to see a positive MR test? Methyl red. You have to write methyl red. You cannot say MR. Okay? What is the pH of a positive test?
Low, acidic.
Huh? It's acidic. Very good. This test is part of some battery of tests. What's the battery? IMVIC. Because it's used for identifying something enteric bacteria. This is a gram specificity. Is it gram positive or gram negative? Only gram negative. There are no such things as true enteric gram positives. They're intestinal bacteria, but we don't, well, I guess we might, but it's mostly gram negative enterics. Okay? What is the primary substrate in the MR or the VP test? Glucose. Okay? This is the VP portion, right? So what does a positive VP test look like? It's red, but it's very, very deep red. It's almost a burgundy. Okay? So if the organism tested positive for VP, it would test MR positive or negative. If it tested VP positive, it would test MR positive or negative? Negative. So, hey folks, listen up. Write the word positive or negative. Don't put a minus or a plus sign because your minuses and pluses look alike. Okay? What reagents are added to the VP test? Potassium hydroxide and naphtol. And what is a catalyst? Creatine. Good. Anybody have any questions about the MR or the VP? Okay. This is the indole test by itself. Okay? So describe a positive indole test. What does that mean, Doc?
A red
The bright red.
It's got a red lake. Yep. Indole is the end product of the digestion of which amino acid? Gobble gobble. Tryptophan. Right? Tryptophan. And a positive test is a positive for which enzyme? Add ACE to tryptophan, you get tryptophanase. Okay? What reagent is added to the tube after incubation to elucidate a positive test? What's the reagent? Kovacs. Kovacs reagent. Okay? Good? Alright, here we go. So this is a nitrate test. There's some stuff right here, just listen to me. In the nitrate test, you inoculate the organism into the test, and you come back in 24 to 48 hours. And to step one, you add two reagents. What are the two reagents you add? Sulfonilic acid and naphthalene. If you get a red color, that's positive, but it's positive for what? Nitrite present, nitrate to nitrite, nitrate reduction to nitrite. So those are all very fair answers. The easiest one to do is just to write nitrite positive or NO2 positive or NO3 to NO2, and those all mean the same thing. There are many ways to answer this. You just have to provide a way that is correct. Okay? If you get no color develop, then you go to step two. And in step two, you add a pinch of what?
Zinc.
Zinc. And what does zinc do? Not catalyze. It's a reducing agent. So, if it turns red after zinc, is it positive or negative?
Negative.
Negative. Because zinc reduced nitrate to nitrite, and the reagents were there, and it immediately turned red. If you add zinc and you get no color, then that is positive, but what happened?
Reduced to gas.
Nitrate was reduced to a gas. Gas freed or liberated, something like that. Okay? The other way you can do that is just to simply say this. You don't know which gas, and it doesn't make a difference. It just means that the organism reduced the nitrate to a gas. We don't know which one. We don't know if it's nitrous oxide. We don't know if it's nitrogen or ammonia. It's all gonna. It doesn't make a difference because we're gonna say that the organism was a fast reducer or reduced nitrate to a gas. Okay. Notice I'm not going to ever want you to tell me which organism is associated with these tests. Not yet. It's coming. But you have to do it in steps. I cannot expect you to have all of this knowledge without first understanding the mechanism. You have to understand the mechanism first before you can use it diagnostically. Okay? Anybody any questions? Here we go. So this is the urease test, also known as urea test, right? A positive urease indicated by what color? Fuchsia. You can't say pink because pink is negative, right? Sometimes it looks orange, right? But usually it's kind of off pink or whatever, okay? Why is it turned fuchsia? Because if you break down the urea, ammonia is released into the environment. Anytime ammonia is released into the environment, the pH goes up. Therefore, the pH indicator being phenol red will turn fuchsia. Okay? The pH indicator in the medium is what? Phenol red. And that is based on pH. What is the reaction there? No, it's either acidic, basic, or neutral. basic, or alcohol, but they mean the same thing. Okay? What's the pH of that guy? It's orange. Neutral.
Neutral.
The end products of your breakdown are, we talked about this already. What are the breakdowns? What are the end products of the breakdown of urea? What are they? Ammonia is the big one, that's the one we want. The other one is a gas and it gets freed. What is that? Carbon dioxide, CO2. Okay? Anybody have questions about urea? Okay. So this is triple sugar iron, right? What is TSI? Oh, I just give you an answer. I have to be careful with that because I give answers left and right. So TSI stands for triple sugar iron. Iron is the presence absence indicator, right? Just like it is in the SIM test, the presence absence indicator is iron. Be sure you know that. The big word for iron is ferrous ammonium sulfate. Or you can just simply say iron. I'm okay with that. Name the 3 sugars that are in the medium. Glucose, lactose, and sucrose. Which one of them is there at a lesser concentration? Glucose. It's there at a 10%, where the others are there at 1%. Okay? How do you inoculate a TSI slant? SSSB. Is the SB really important? Yeah, because as we found out today, there were some people who were light butt stabbers or didn't stab the butt at all, and you couldn't see the full result. Okay? And you interpret that by putting answers in between the 2 forward slashes. So now you have to listen to me, right? If you are taking it in an alternate format, I've had enough description in it that it should be there. But in the classroom, I'm gonna say it's yellow here and it's orange there. And you're gonna say, what here goes what? A. And no gas. Why? Because gas can only be produced in acidic conditions. If it's neutral, it cannot have any gas.
So that's only based on the butt, on the answer on the butt.
The gas?
Uh-huh.
That's correct.
Oh.
Only on the butt.
Only those.
That's why the stabbing of the butt is really important. Okay? If you're a light stabber or didn't do it at all, you're not gonna get the full effects. Okay, let's go on. So here, I've got fuchsia here and orange here. Go.
Fuchsia is.
Fuchsia. So what is it? K. And no gas. Okay? Jeanette, you're making a face. Why are you making a face?
What was my thinking?
So you have to listen to me. Remember, this is a time test this time. You won't be able to go back at the end. You have to ask the clarification questions during the time we're taking the test. We will not be going back. Yes, Malika?
So is it K no gas?
K and no gas.
Oh.
You have to listen to me because I'm going to describe it. Because it's really hard to see there's a big difference here, but there is a difference. Okay? All right, here, let's do this one here. So I've got fuchsia there and I got black there. Go. K, A, hydrogen sulfide. What about here? I've got fuchsia here, I've got yellow there, and I've got cracks and fissures. I've got broken media. Go. K, A, gas, just gas, not hydrogen sulfide. Okay? So what is added to the medium to give the black precipitate? Or the black color? Ferrous ammonium sulfate or iron. I-R-O-N. Okay? I'm not going to ask this question because I did not talk about it. So I will not ask the questions that have not been talked about. Okay? Alright, so let's just practice real quickly because we have some time. So look what's going on, what craziness is going on here, right? So this is somebody who stabbed it and then went all the way through. So you have this thing here. So I got what here? A gas. Let's do this one. I got cracks and fissures. I got what? A gas. Very good. What about here? I got fuchsia there. I got black there. Go. K.
And
No, it has to be A. Because the rule is gas can only be produced at acidic conditions. That's hydrogen sulfide. It is a gas. Therefore, this is KaH2S. Okay? Good. So this is the DNase test. Okay? Why is DNase called a ribonuclease? Because it can break down a nucleic acid. Okay? What is the indicator in the mean? This is the presence-absence indicator. Methyl ring. Very good. And describe a positive test. What's a positive test look like? Clear around the colony. Mm-hmm. Which one is clear around the colony? Serratia marcescens or Staph aureus? Serratia marcescens. Although group 6 kind of makes up their own rules over there. Okay, so what's the major substrate in the MR? Good, glucose. What's the major substrate in the nitrate reduction test? Nitrate. And what's the major substrate in the VP? Glucose. I did that for a purpose. If you know the name of the test, a lot of times you know the substrate. MRVP you don't. It doesn't tell you. But glucose is a major substrate there. Okay? This is a gelatin hydrolysis test. What's the substrate? Gelatin, very good. Define substrate.
it's what is used by the organism.
It's what is being used as a nutrient or what's broken down or what's being acted upon. Very good. What's the purpose of placing a tube at 4 degrees Celsius? Refrigerator.
To confirm whether gelatin has been hydrolyzed rather than melted.
Solidified for the control tools.
You too could be a microbiologist.
I'll consider it.
Which one is negative, A or B? B. That's liquid, so it's positive. Okay? All right, here's the urea test. So, didn't we list this one already? No. So, what's the result of tube C? Positive. Write the word positive. Don't put plus or minuses. Got that nice? Everything okay? See you Thursday. And then, what's the pH of tube A? It's orange. Neutral. Very good. Okay, so I don't know why this is in here again. It's just written a different way. Okay, but here, what is the result of a negative MR and a negative VP? Alkaline, that's correct. What is the result of a positive MR and a positive VP? You made an error. What about a negative MR and a positive VP? What does VP test for? Neutral end product. So if you have a positive VP, it's neutral. What about a positive MR and a negative VP? It's acidic. We did nitrate already. Here's the carbohydrate fermentation test, also known as the phenol red broth test. Okay, so read this. This is it sees the control. It's uninoculated. Identify the test which shows a complete fermentation. A, B, C, D, or E? A, right? You gotta have gas and acid. Identify which tube shows an acid production only. B. Identify which tube shows alkaline byproducts or end products only.
D.
D. What's the pH indicator? Phenol ring. phenol red. Remember, if you don't know the name of the pH indicator, guess phenol red. There's only 2 pH indicators. So really, every time I ask that, it becomes a multiple choice and it's 1 or 2. It's either phenol red or bromo-thiamethyl. Because those are the only pH indicators we've used this time. There's more coming. But those are the 2 that we've used so far. Okay? So then there'll be some lab. I've already asked some of these, right? So in hazardous communication, in the blue area, what type of a hazard is that communicating? Health or toxicity? What type of information is found in the SDSs? Chemical information. Okay. Where's the rally point? Yeah, they should be Riverside, not HOC. Where's the rally point for Riverside? The golf course, right? So you go down the stairwell, go to the golf course, off the street, so you don't block emergency vehicles coming. If you were to contaminate your gloves with E. coli, where would you place them? This happened today, unfortunately. Huh? In the biohazard bag. What tool or utensil is used to inoculate SIM? Needle. And in our lab, where does one place tubes or growth plates so that they are maintained but do not continue to grow? Which tray? Blue tray. Okay. Rachel, we get the lights. So, last year I went to a conference and Dr. Anna Maria took my class one day. Are there any questions about, yeah, it was on?
The start hydraulics test, there was a question on it?
I didn't have a question on it, but it may be there.
Oh, cause it was in our,
And the lab review.
on the lab review. The slide.
The second to last.
Oh, it
slide.
is?
Yeah.
I had a question on it.
Look. Let's see what I got. Oh, cause I did that for HLC, okay, fine. I'm gonna get that faster.
Yep, that one.
So, there are several in here, so let's just do starch. So, what's the substrate of the starch hydrolysis test? Starch. What reagent is added? Iodine. A or B, which one's positive? B. And then this is skim milk agar. So, what's the substrate? Huh?
Oh wait,
Casein. You can't say skim milk, because skim milk also has lactose in it. And we're looking specifically for the enzyme that breaks down casein. And which one is positive, A or B? A. B is a little bit positive also, but A is really positive. Okay. And then lipid hydrolysis. What's the substrate in the lipid hydrolysis? Tributylin. You could also say a lipid. I'm okay with that.
Okay.
And which one is positive, A or B? A. And then we talk about That was the only difference. Okay. Anybody else have questions? What time is the lab practical?
10:35.
It's gonna rain tonight, but it probably won't rain on Thursday, so you have enough time to get here, have a taco.